Hello everyone, and welcome to today's live broadcast, CE-SELEX, Capillary Electrophoresis Selection of Aptamers, presented by Dr. Michael T. Bowser from the University of Minnesota. Today's webinar is sponsored by SCIEX, a global leader in life sciences and analytical technologies, offering innovative breakthrough in mass spectrometry, liquid chromatography, and capillary electrophoresis. We at SCIEX are excited to bring you another in a series of educational web seminars via LabRoots, the leading scientific social networking website and provider of virtual events and webinars, advancing the scientific collaboration and learning. I'm Alan Chanity, Senior Product Manager for the PACE MDQ Plus Capillary Electrophoresis System at SCIEX Separations, and I'll be your moderator for today's event. But before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button. It's located in the lower left of the presentation window. And then type your question into the box that appears on the screen. We will answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. And to enlarge that window, just click on the screen icon located on the lower right. If you do have any trouble seeing or hearing the presentation, please click on the support button at the top right of your presentation window, or use the Q&A button to let us know that you're having a problem. Now remember, this is an educational webinar and thus offers free continuing educational credits. So after the webinar is over, click on the CE button located in the bottom left-hand corner of your web page and just follow the process to obtain your credits. Now I'd like to introduce Dr. Michael Bowser, Professor of Chemistry at the University of Minnesota. Dr. Bowser received his PhD in David Chen's lab at the University of British Columbia and completed his postdoctorate in Robert Kennedy's lab at the University of Florida. Using capillary electrophoresis, Dr. Bowser and his group are developing faster and more effective selection techniques to discover protein or peptide binding DNAs and RNAs that can be used as therapeutics, diagnostics, or catalysts to help solve challenging biological problems. Now, along with Aptamers, the Bowser Biotechnology Laboratory's interests also include developing enabling technologies in microfluidics and microdialysis that promote advances in biological and chemical sciences. So please join me in welcoming Dr. Michael Bowser. I will now turn over the presentation to him. Michael?
Thanks, Alan. I'd just like to start off by thanking SCIEX for giving me the opportunity to share some of our work with all of you. We've been working for a long time in developing new and faster ways for finding these aptamers that a lot of people are using for pharmaceutical development and diagnostic development. So let's just start off with, what are aptamers? Aptamers are going to be single-stranded DNA or RNA molecules that can kind of fold into some of these interesting shapes. And I've shown an example here of just a two-dimensional kind of representation of one of the aptamers that we've developed in our lab. This one actually folds into a shape that allows it to bind neuropeptide Y. So normally when we think of DNA and maybe RNA, we think of sort of that double helix. We see that pretty picture put on, the journals and magazines and things like this. From our perspective, that's actually a pretty boring structure. When you make DNA or RNA single-stranded, you have these opportunities for these stems, these loops, these knots, these quadruplexes, all of these other different kinds of shapes, which actually starts to turn the interesting parts of the chemistry of the nucleic acid out, whether it's hydrogen bonding sites, whether it's ionic bonding sites, whether it's aromatic group stacking, things like that. What that does is it gives an opportunity to make binding pockets or binding sites that can bind proteins, small molecules, other types of targets with high affinity. And that's really what aptamers do. They're these nucleic acids that we found that make a particular structure that can bind that target both with high affinity, high selectivity. Very often they're compared to antibodies because they have very similar properties. When we're talking about high affinity, the best aptamers are binding with low nanomolar to high picomolar dissociation constants. Very often they're thousands of fold more selective for their particular target than similar kinds of other molecules. Other advantages of aptamers include that they're generally non-toxic and non-immunogenic. They're going to be nucleic acids, so they're things that are readily, accessible to the body and are non-toxic to the body and generally don't, generate an immune response. The pharmacokinetics of aptamers are very predictable, again, because they are going to be nucleic acids, and if you have one particular aptamer, you know the pharmacokinetics of that. If you make another aptamer, the clearance mechanisms, the degradation mechanisms, the delivery mechanisms are all going to be fairly similar. You can prepare aptamers using solid phase synthesis. You can prepare them in bulk, somewhere around $200 to $2,000 a gram. If you keep them dry, they actually have a very long shelf life. They're very accommodating of changes in temperature. They're much easier to kind of store, ship than proteins or other biologics. You can develop aptamers that target very toxic compounds or things that are on the other end are very non-toxic and don't generate an immune response. These are difficult things to get antibodies to generate an immune response against. If the target is so toxic that you can't deliver it to an animal, it's hard to generate an antibody for that. One of the interesting things in biotechnology is that it's pretty easy to unfold aptamers, either by putting them in denaturing conditions or heating them up, and then bringing them back into renaturing conditions. Very often they'll fold into the correct shape again. And so that opens up some interesting technology applications where you can bind a target, release a target, and then regenerate that binding surface again. That's pretty tricky to do with protein binders and things like that. Also, it's pretty easy to think about what the antidote generation is or to be changing binding sites because you can just look at the complementary sequence of that aptamer or that nucleic acid and you can start to change the folds or change kind of the shapes of those aptamers. So that's all good. But some of you are probably thinking that there's lots of different types of molecules or proteins or things like that that have a lot of those same properties that I just described. And what really makes people excited about aptamers isn't so much what they can do, it's that we have kind of a unique process for finding them. And it really boils down to combinatorial chemistry, and so much of chemistry is based on finding binding or finding interactions, and there's lots of different ways we can go after this. And in combinatorial chemistry, what we're going to do is we'll make a large pool of molecules and try and pick out those ones that have that correct binding shape or that binding interaction with that target we're looking for. The CE-SELEX process, also called in vitro evolution or in vitro selection, was developed in the early 1990s by Larry Gold's group, Jack Shostak's group. Andy Ellington was an early pioneer in this. What it does is it takes an alternative approach to the combinatorial problem. And what we're going to do is we're going to take a random sequence library of DNA or RNA, and we're going to take about 10 to the 13, 10 to the 14 different random sequences. And we can synthesize these, and we're going to put a 20 base primer, a 40 to 60 base random region in the middle, and then another 20 base primer at the other end. And that's going to be our combinatorial pool. And then the trick is what we have to do is find which sequences in that combinatorial pool fold into the correct shape to bind our target. So what we're going to do is we're going to allow it to incubate the target, and then we're going to have some kind of selection procedure that's going to be able to pull out those sequences that take the right shape. Now, when we're looking at a protein target, the most common way to do this is with nitrocellulose filtration. What we'll do is we'll take our random sequence library, we'll allow it to incubate with the protein target, run it through nitrocellulose. Nitrocellulose is going to bind proteins, it's going to pull down the protein targets, and if any of the DNA or RNA stuck to that, it's going to be retained on the filter. Then we can wash everything else off to waste. Then we're going to basically take our sequences that are retained on the column. Because it's a nucleic acid, now we can think about making more of that nucleic acid. And that's why it's really important to use nucleic acids in this process, because if we start off with 10 to the 13, 10 to the 14 different sequences, maybe only 1,000 of those fold into the correct shape to bind our target. We can use PCR amplification to make a lot more copies of that sequence again. Now, PCR isn't perfect. It's actually going to put some mutations or put some changes into that when it's making the copies. And so we're actually introducing some more variability back into the pool. And then we cycle back again, and that gives us a new pool after we purify it and make it single-stranded and to be able to repeat the process. Some people refer to this as selective evolution, in vitro evolution, because what we've done is we've taken all of the steps that we see in nature, whether it's some type of selection criteria, whether it's reproduction, which is the PCR, and then mutagenesis, and then repeating that. And as we go around the cycle again and again, we're actually saying that the pool is evolving to bind to the target. Now, when we typically do this using nitrocellulose filtration or affinity chromatography to pull down those binding sequences, we're thinking about going around this process 10 to 15 times. It might take a day or two to do all of the steps in this process. So we're talking about a process that's going to take weeks, possibly months, but to enrich that pool with high affinity binders. There's other issues kind of as well. We have to think about the challenges of doing the selection on a stationary surface, whether that is with a filter or with its affinity chromatography. You really have to worry quite a bit about nonspecific binding. And when you're doing nitrocellulose filtration, you have to actually do negative selections where you run the pool through the nitrocellulose first and pull out any of the binders that just have background affinity for the nitrocellulose. It's very common to go through this whole CE-SELEX process, 4 weeks, 6 weeks, go back, test your aptamers, and you found you've made aptamers for nitrocellulose. You want to make sure you don't do that. So it would be nice to be able to take away some of those stationary supports. Also, if you're going to use some other kind of affinity procedure, whether it's affinity chromatography or a panning process, you think about those best binders attaching there to the surface. You have to think about a way of getting them back off the surface. And it may be actually easier to get some of those binders, but not the strongest binders, off of that surface. And so the kinetics of getting those sequences back off the surface may actually work against you. So almost 15 years ago, our group introduced a new approach to selecting these aptamers. And what we did is we decided we wanted to use capillary electrophoresis and replace that nitrocellulose filtration or affinity chromatography. Capillary electrophoresis, what we're going to do is we're going to take a thin fused silica capillary, maybe 50 microns in diameter. We're going to fill that with a buffer solution. We're going to load our sample into there, and we're going to apply an electric field across that capillary, maybe 20 to 30,000 volts. Then the molecules are going to move in that electric field depending on their charge and size. Now, capillary electrophoresis has been an enabling technology in genomics and using gel electrophoresis to separate DNA based on size. We don't want to separate DNA based on size. We want to separate it based on its affinity for the target. So what we're going to do is, instead of putting a gel in the capillary, we're going to just put a regular buffer solution in and do free zone electrophoresis. When you do free zone electrophoresis, all of the sequences that don't have affinity for the target, it doesn't matter what their size is, doesn't matter what shape they are, they're going to migrate at a certain position or a certain time through the capillary. Any of the sequences that bind to the target, it's going to change their size, it's going to change their charge, and they're going to migrate through the capillary at a different velocity. And so what we're going to do is we're going to fraction collect the binders from the non-binders, and that's going to allow us to separate out the aptamers that we want. This is what one of our first capillary electrophoresis separations look like. Every time I put this up, I cringe a little bit because this is not normally what capillary electrophoresis looks like. Capillary electrophoresis usually has these very narrow peaks, high efficiencies, 100,000 plates, very fast separations. But we're using this separation a little bit of a different kind of way. We just really want to separate two things, binders from non-binders. And so what we did here is we injected a pretty large number of sequences onto this small volume. One of the challenges we have is normally in capillary electrophoresis you're going to inject a couple of nanoliters onto that column and then do the separation. But we want to inject as many sequences as possible because we want to increase the chances of there being a high affinity sequence in that pool. So we trade this off by really over-injecting in this case here, and we're getting this very broad peak. It's a pretty high concentration of DNA, but we're able to load close to 10 to the 14 different sequences onto the column in this case. Now, although the peak looks kind of messy, there's a kind of a unique thing in this selection that we're doing here. Here we're going to do a selection, and we're going to try and find an aptamer for IgE. Turns out that IgE is a very large protein target. It's slightly positively charged. When you bind the DNA to that, it essentially stops in the capillary. And so the DNA, free DNA is coming out here. The actual DNA IgE complex is actually going to be way off the screen over here, somewhere about 20 or 30 minutes out. And so although it looks like a messy peak, we're actually doing a really good job here of separating the binding sequences from the non-binding sequences. This slide here is showing some of the things that we're thinking about when we're doing the fraction collection. And so, again, we're going to be able to see in this early round a relatively large peak here that corresponds to the unbound DNA. Most of the sequences are not going to bind. We're going to want to put a lot of sequences in, again, because we want to increase the chance that we have a strong binding aptamer in there. On the other hand, early on, we have very, very few sequences that can bind the target, maybe 1,000, maybe 10,000. And even if we have those bind the target very strongly, there's just not going to be enough of them to see a big peak. And so when we're doing our fraction collection, very often we're doing it blind. We will not see a peak for that aptamer protein or aptamer target complex. So what we're going to do is we're going to take a look at that non-binding peak, and then what we're going to do is we're going to say, let's go a certain distance after the peak here and say we're going to have a window after that where we expect to find our complex peak going to be in. And we can do this by doing some preliminary experiments here to say, okay, let's figure out where DNA is going to migrate. Let's run our target. Here, in this case, we're doing a selection against neuropeptide Y. We know that our complex is going to be somewhere in this region here in between the protein target and the free DNA. So in this case here, we've decided to cut off our fraction collection where we're showing that arrow there kind of in the middle. On the right here, we're showing the case here where we've coded the capillary here. And so in this case over here, we're not going to have any electroosmotic flow. We can think of DNA moving through. It's going to have a very negative charge. We're going to have a negative charge at the inlet of the capillary, and it's going to be the fastest thing that moves through. As soon as we bind a target to that DNA, it's hard to imagine many targets being as highly negatively charged as DNA. So we normally think about binding the target and it's slowing it down, and that's why we have this collection window after the DNA over here. On the bottom here, we're showing kind of how the on-column detection is going to work in capillary electrophoresis. We can see that peak over here move past the detector, and then we have a certain amount of time before it makes it all the way to the outlet of the capillary where we're going to do the fraction collection. And so we can actually do this fraction collection and calculate the time window that we want to do the fraction collection on the fly. And how far after the non-binding sequences we cut things off and start to collect the sequences looking for binding sequences is going to depend on how big of a mobility shift we expect. And it's going to depend on that particular target. So in the example here, we're showing a window that's about 1 or 2 minutes after the unbound DNA comes off. We stop the separation, change the vial, collect any sequences that are left on the column into a different vial that's going to make it ready for PCR. If we had a smaller target, we might narrow that window and move it closer to the unbound DNA. And there's a trade-off in doing this. The closer you get to the unbound DNA, the more non-binding sequences you're going to catch in that tail. But on the other hand, you have a better chance of collecting those binding sequences. Now, coated capillaries are great if you're going to have a protein target that has affinity for glass or is going to stick to the glass surface. Very often you want to just run a separation, kind of like what we're showing here with the neuropeptide Y, where we are going to just make sure that that protein target over here doesn't have affinity for the glass. We're not getting tailing, we're not getting streaking, things like that. But in other cases, you're going to have a protein target that does have affinity for the capillary column, and then you want to make sure you use one of these coded columns. If you run that and your protein target behaves well on a bare fused silica capillary, you can run it on just a regular uncoated capillary. Use an uncoated capillary, you're going to have electroosmotic flow that's going to reverse the order of all the peaks that are going out. It's going to pull everything towards the anode, or it's going to pull everything towards the negatively charged electrode. What's going to happen is the migration order for everything is going to reverse. And now we're going to have the binding sequences are going to come out before the free DNA over here, because we've shifted the electrodes on our capillary. Now it becomes a little bit trickier to do this fraction collection, but because, again, we're doing this on-column detection, we can actually wait until this leading edge of the peak over here reaches our detection window, and we still have a lot of those complexes still on the column. And so what we can do is now we're going to migrate all the binding sequences into a collection vial at the end. Once we get to this point over here, that's 1 or 2 minutes before the leading edge of our non-binding sequences. Once that reaches the outlet of the capillary, we're going to stop the separation, and it's basically going to be done. This again is showing where we've basically cut things off doing our neuropeptide selection here. I will point out here that this separation here in the blue trace was actually collected using fluorescence. In our earlier data, we very often use absorbance detection. We found more recently that fluorescence detection makes it much easier to see these peaks, much easier to do this fraction collection. Here we're showing how we're doing the fraction collection for one of our selections against trying to find aptamers for HIV-RT. This is a little bit different than our IgE selection, where the resolution between the non-binding sequences and where we expected the complex to come out was so large. Here we're doing an uncoated capillary, so again the complex is going to come out before the non-binding sequences. And when we do this large injection like we did with the IgE selection, we get, again, this really broad peak, and the peak is leading so much that it's leading out over where we expect to find the complex take place. And if we try and do the fraction collection this way, we're going to have a lot of these non-binding sequences be collected at the same time point where the complex is going to come out. It's going to be very difficult to do the selection. So in this case here, what we found is that we actually have to do a trade-off. By decreasing the number of sequences we're injecting, we actually start to get better peak shape, and we start to get better resolution between the complex and the non-binding sequences. So there is a bit of a trade-off on how you're developing kind of these separation conditions at the beginning. After we go through and do this process and do the fraction collection using capillary electrophoresis, and again do the PCR to make more sequences, make them single-strand, purify, and repeat the process, what we want to do is make sure that the pool is actually evolving and we're actually selecting that pool to bind our target. So in this case here, we're doing absorbance detection, and we're looking at a peak here for the non-binding sequences over here, and you'll notice that that peak intensity is quite a bit lower than some of the earlier data that I showed, and the reason for that is because during the PCR amplification we can only get that concentration of the DNA up to sort of low micromolar concentrations, and that's right around that detection limit of the absorbance detector on our CE instrument. Again, if you do this with fluorescence detection, the sensitivity is quite a bit higher and it gets a little bit easier to see this peak. Now what we did in this experiment here is we titrated with increasing concentrations of our target, and we can actually see that the free peak here drops in intensity as we're adding that IgE. And that's showing us that as we go through different rounds of selection here, in this case this is just after a single round of selection, we're able to find that about 2/3 of our pool is already able to bind IgE. Instead of just looking at the amount of the pool that can bind, we think it's a really good idea to actually track what the dissociation constant is of the pool as you're going through different rounds of selection. This isn't a true thermodynamic dissociation constant because we're looking at a collection of different sequences here, but it does give us an idea of how the selection is progressing as we go from round to round. In this case here, again, this is one of our IgE selections where we're trying to find Aptamer against IgE, and we see after a single round of selection, the average dissociation constant in our pool is already about 15 nanomolar, which is really good. You'll see here as we went through later rounds of selection, the dissociation constant actually seemed to get a little bit worse before it reconverged back onto this low nanomolar dissociation constant over here. And I'm going to talk a little bit about why we think this is taking place. This is not an uncommon thing for us to see as we're going through different selections. So normally what we would do, we would run through several rounds of selection, we would come back, measure the affinity of the pools, and then we would come back and after we see that the affinity of the pool isn't improving anymore, we would pull those sequences out for cloning and sequencing. So again, it's really important to note that as you go through this process, you're always going to have a collection, a mixture of different sequences that are going through. You're never going to boil this down to just one or two different sequences. You're always going to have a pool. So after you've converged the pool as much as you can, again, you're going to stop. You're going to want to sequence, clone sequence, and synthesize individual sequences for further analysis. We did this with our IgE selection. You get this long string of sequences back. If you're doing conventional cloning, you'll have somewhere between 30 and 100 different sequences to look at. And what's commonly done in the CE-SELEX community or the Aptamer community is to go through and look for these sequences and use different software programs to find motifs and to find patterns that exist between the different sequences we come back. And the two sequences that I have on the slide right now are actually an anomaly in our research. This is one of the very few sequences where we've found a motif when we've gone back and looked at the cloning data. Through all of the clones we looked at through IgE doing the selections many times, our HIV-RT selections, our neuropeptide Y selections, all the other selections we've done, usually we come back with the capillary electrophoresis selections and we don't find these motifs. Usually we go back and we look at the sequences and we can't find any patterns. We can't find any kind of at least immediate structural comparisons between this. That doesn't mean that they're not making the same kinds of structures. It's just not obvious in the sequence. Once you have these sequences back, you need to come back and measure the affinity of these sequences for the target here. So what we've done in this case here is we've taken the sequences we came back. This is one of the clones here from the previous slide, so this is one of the ones where we found this motif for. We put a fluorescent label on this, so now we're actually getting nice signals here on the capillary electrophoresis. And this top peak here is showing the intensity of the peak when we don't have any IgE in. And again, we're titrating with increasing concentrations of IgE here, and we see that free peak drop. We can use that to calculate how much the fraction of the aptamer that's bound at different IgE concentrations, and we get sort of that typical rectangular hyperbola here, and we can fit this and get a dissociation constant. It's really critical here to use the laser-induced fluorescence detection for this because you need to be performing this experiment at low concentrations here to be able to get down to the dissociation constants and measure the dissociation constants that are really typical with aptamers that you're going to get. So we did this. We looked at a number of clones. Again, we had trouble finding motifs. We didn't really find too many motifs in the clones we found, and so as an analytical chemist, we went back and we decided we're going to randomly sample the pool. And so we actually decided, instead of trying to be clever and try and find sequences that we predicted to have high affinity, we used a random number generator to actually pick these out. And we want to just get a sampling of the pool and just see if we were just to pick sequences out, what kind of dissociation constants we would get. And on the slide right now, we have 6 different ones that were chosen at random. These were the only 6 that we tested from this, so I'm not cherry-picking the data here. And we're showing on the right side here what the dissociation constants we measured for these clones were. What was really surprising to us was how tightly clustered these dissociation constants were, because again, we're just kind of picking these at random. All the sequences we picked out bound, all of them bound with high affinity, but they're all tightly clustered around that 30 nanomolar value. And we're showing down here in the bottom actually what the standard deviation as distribution is. We think this is really interesting because, again, we're not finding motifs, we're not finding a lot of patterns, so that means we're finding a lot of different sequences. But we add to this that all of the sequences that we're pulling out of that pool, even though they look really different, they're all binding our target with high affinity. And so what that tells us is that we have lots of different options to pull out of this pool to find sequences with high affinity for our target. A couple of cautions when doing selections. A couple of things to think about. One of the first things that you should do is measure the affinity of just the random sequence library for your target. I've seen many times where people have spent weeks or months doing their selections. They come back, they find sequences that bind to their target with maybe high nanomolar dissociation constants. They're very excited about this. They go back and they check against the library and they find the library binds just as well. Okay, there's lots of proteins out there that have nucleic binding sites on them that have sort of non-specific kind of interactions with nucleic acids. So you want to make sure that you're actually improving the library, improving over the library for the affinity for the target. So in this case here, we've just took the random sequence library, we measured the affinity against IgE, and we're getting a dissociation constant somewhere around 2 micromolar. So we're talking about improving the affinity of our pool almost 100-fold over the affinity of just that random sequence library. We think it's really important to go back when you're characterizing your aptamers to use more than one technique to measure the affinity for your target. Measuring affinity for targets or measuring these kind of binding interactions can be very tricky. It's very easy to introduce biases, whether that's changing viscosity, whether it's changing pH, whether it's changing ionic strength, and getting curves that look like these rectangular hyperbolae. So we think it's really important to measure with at least 2 different techniques to make sure that you really are getting affinity is what you're measuring. In this case here, we're showing an ultra-filtration kind of approach where we've either used fluorescence or radio labeling. People use dot blot. We like using fluorescence polarization a lot. But it's important to find a couple of different complementary techniques so when you do see binding, you feel really confident that in that binding. The slide here is comparing the measurements that we're getting with affinity capillary electrophoresis with our ultra-filtration data, and we can see that the dissociation constants line up very well, and actually this is fairly reproducible across the different techniques. So again, this adds confidence in our measurements here. The other thing I'll point out is when we start to average the dissociation constants between 2 selection rounds and 4 selection rounds, we see that there really isn't that much improvement by going those additional rounds. And so within 2 rounds of capillary electrophoresis selection, we're able to get this pool to have a lot of these sequences that have this low nanomolar affinity for, in this case, IgE. We want to think about selectivity. How selective is your aptamer for your particular target? So in this case here, we're looking at comparing our affinity for IgE versus human IgG. In this case, we went up to 20 micromolar. We're not really seeing any affinity. So we're showing good selectivity for human IgE versus IgG. Here we're looking at the same protein, different species. We only went up to 2 micromolar because Mouse IgE is quite a bit more expensive, but we're not seeing any affinity up to 2 micromolar. So again, we're seeing good selectivity for human IgE versus human IgG or Mouse IgE. So it just really reinforces the selectivity that we can get for aptamers. One of the important things in this selection is we didn't put anything in the selection criteria that specifically made the aptamer selective for human IgE. It just very often as a result that enabled to get to this high affinity interaction, you need to have lots of different binding sites on the target, and that basically gives you selectivity out of this as well. The slide here is comparing our capillary electrophoresis SELEX versus other selections that have been done in the literature. What we'll notice from this slide is the dissociation constants that we get are very comparable to those that have been previously demonstrated. What the really big difference, though, is the amount of time and the number of rounds of selection that it takes. Conventional CE-SELEX, again, is very typical, 10, 15 different rounds of the CE-SELEX process, and again, taking weeks, possibly months to do. With capillary electrophoresis, we're finding that we get aptamers usually within 2 to 4 rounds of selection. So instead of it taking weeks to months, we're talking about getting through this process within a week if it's taking us about a day to do 1 round of selection. So to finish my talk, I just want to run through a couple of, kind of the unique things about capillary electrophoresis, a couple of the challenges that we have in some of the work that we've done to try and understand what's going on in some of the things that are maybe a little bit different than conventional selections. One of the initial concerns we had with capillary electrophoresis was how small of a target could we look at. All of the selection is based on getting this mobility shift here in the capillary, and if the target isn't very big, is it going to change the size or is it going to change the charge enough for us to be able to move that complex far enough away from the non-binding for us to be able to do this fraction collection? And so we've been able to do selections against neuropeptide Y. Neuropeptide Y is about 1/4 the size of our DNA library. But we really wanted to push things, and we decided let's go for a small molecule target. And we decided to go after this N-methyl mesoporphyrin and NMM. Okay, it's a molecule, only a molecular weight of 580. And again, to compare that to the typical molecular weight in the DNA library, those molecules are about 25,000. So it's quite a bit smaller than the DNA. And we wanted to see, would it even be possible for it to change the mobility of the DNA enough for us to be able to do effective fraction collection? Here showing us doing the fraction collection. Again, we're doing absorbance here, so the signal in the second round isn't great. It would be a little bit easier for doing this in fluorescence. And the arrow here is showing where we're cutting things off. Again, we're going on the leading edge here of the non-binding sequences, and we're cutting this basically as close as we can because we're really not expecting a very big mobility shift when it binds that small target. In the later rounds, again, we're getting a small peak because we can't put in as many sequences. We don't generate as many sequences when we're doing the PCR amplification, but it is big enough here to be able to see where we're going to do that fraction collection. We took this through and we ran it through, in this case here, 6 different rounds of selection, and we looked at a control, we looked at a library here, and what the assay that we're using here to track this is we're actually following the fluorescence of the porphyrin itself. When it binds the target, it changes the environment. We're expecting the fluorescence to change, and we can see that over 4 rounds of selection, we're starting to see that fluorescence increase here as we add the pool to it. And so we were thinking that this is increasing the affinity for the target. The slide here is comparing the dissociation constants, the binding curves here, for just the random sequence library to one of our aptamers after 6 rounds of selection. And you can see really clearly here, based on how sharp the curve is here, that we get binding at much lower concentrations of DNA with our selected clone than we do with the library. And so we show we were actually able to get affinity for the target. The affinity dissociation constant here we're getting is about 1 micromolar. That's pretty good for small molecule targets. Very often you don't get affinities quite as high. This table here is showing sort of the dissociation constants we got from some of the different clones. We saw that after 3 rounds of selection, okay, and so that's what the 3s are going to be over here. We're able to get, a number of aptamers here with low micromolar dissociation constants. And we saw a strange thing here is that after 6 rounds of selection, the pool actually got worse. And this actually matches up with the assay that we were looking at with the pool here, where we're actually seeing the fluorescence decrease as we increase the number of rounds of selection here. And so that was a little bit surprising to us. But after 3 rounds of selection, we were able to actually get this to work, and we were able to get these aptamers with this low micromolar dissociation constants. Now, one of the unique things about this N-methyl mesoporphyrin is that it it is actually a transition state analog for this porphyrin metallation reaction that's shown on the screen right now. And so if we take the porphyrin here, putting the methyl group in the middle actually wobbles the porphyrin a little bit, which mimics the transition state of inserting this copper ion into the porphyrin here. And so the reason people select antibodies or aptamers for this particular target is they want to see if they can get something that actually catalyzes this metallation here of the porphyrin. We're actually able to show that we're able to do this. Well, the way you can track this is by looking at the absorbance spectra over here. And you can see that when we look at the absorbance spectrum, when we put the copper into our porphyrin over here, we get this emergence of these bands at about 550, 570. And we can track the absorbance over here to actually track the metallation of our mesoporphyrin over there. So this slide here is just showing the kinetics of this metallation, and we're comparing the rate of the reaction here with just a random sequence library, and here showing the rate of the reaction here with one of our selected clones, this clone after 6 rounds of selection. Now, it didn't improve the kinetics by a lot. It sped the reaction by about a factor of 2-fold, but it is a statistical difference, and we showed that we're able actually to get aptamers that not only bind to the target, but actually can catalyze this reaction. Again, we think it's really important that we're able to show that we're even able to get something that binds a small molecule target. The way we think this is working is not so much that it's the size of the target that's inducing this mobility shift, is that the sequences, when they bind to the target, they have to fold around the target, and that's actually changing the shape of those DNA sequences, and it's changing how they migrate through the capillary. Now, one of the other questions we have is that one of the nice things about capillary electrophoresis selections is we're getting these high affinity aptamers within 2 to 4 rounds of selection. One of the problems, though, is as we go to even more rounds of selection, instead of the pools getting better, very often we find that they get worse. And we were very surprised about this. We're also wanted to get more insight into why we don't get these motifs emerge and why we get so much heterogeneity in the pool after CE selection, and that, again, that's different than regular selections that are out there. And so what we wanted to do is we wanted to come back and do high throughput sequencing and look at basically the composition of this pool here as we go through multiple rounds of selection here and see how that is evolving and actually be able to track sequences through this. So we did a selection here against VEGF, vascular endothelial growth factor. It's a very common target for selections. In this example here, again, we're looking at the affinity progressing through different rounds of selection, and we can see the library has very low affinity, and within 1 round of selection we're able to get sort of this low nanomolar dissociation constants again. Very typical of capillary electrophoresis. Again, what we're doing here is we're going sort of past that optimum point and doing more and more rounds to basically see what happens to the pool as we go even further. The slide here is showing basically the number of sequences. We used high throughput 454 sequencing to basically do this. Instead of looking at 30 or 100 different clones after every round, this allows us to look at, tens of thousands of sequences after every round. And again, so that helps us look at more sequences and pull out more patterns in this case. When we go and look through this, we can see when we do the sequencing from the library up till about round 5, when we go through this and do the sequencing, we're getting about 30,000 sequences back. And then when we go to later rounds beyond that, we actually see the number of sequences drop off. And I will point out that this is on a log scale, so we're going from about 30,000 to somewhere below 1,000 sequences that we're getting after the round here. Not only that, we're actually seeing that the number of sequences that are the proper length is actually dropping, and that the average length of the sequences in the pool is actually dropping. So we're starting off with sequences that are 80 bases long, and as we're going through the process more and more and more, we're starting to see that those sequences are starting to get shorter and shorter. And that's telling us that something here is going wrong with the selection. So this slide here is actually plotting a histogram of the lengths of the sequences we're seeing in that high throughput sequencing data. And when we look at the initial library, everything looks great. most of the sequences are clustered right around those 80 bases. That's what we expect. That's what we made the library to be. After one round of selection, they're all around 80 bases. And then after 2 rounds of selection, we start to see these smaller sequences emerge over here, somewhere around 47 bases long. These are probably primer dimers or misannealing or something like that. And these basically persist for a few rounds, and then once we get up to round 6 and 7, we see this explosion of sequences that are not the right length. And they're getting up to almost 25% of the abundance of the actual sequences that we want. And we can see as we're going through more and more rounds of selection that something here is not going well with the amplification or with the PCR. When we look carefully at the sequences we see through the different rounds here, what's pretty unique about this and gives us some insight into what's going on is it's not just a collection of different sequences that are all kind of the wrong length. We'll see that almost all of the sequences that are these byproducts, these short sequences that are too small, they're pretty much all the same thing. And so that's telling us that they've originated from a particular misannealing or a particular misamplification that's occurring again and again. And once they start to enter the pool, they persist through the pool. And so that's telling us it's not just sort of a random event, that it has something to do with our primer design and things going like that. And again, even once we get up to these later rounds over here, when we get to pool 7, again, where we're getting these high abundance of these sequences that are not the right length, again, the sequences that we see, the 54, the 46, the 42 long sequences, again, they're all repeatable sequences. And so that's telling us that there's a specific thing, something going on with the primer design here that's leading this misamplification. And so what we really think is going on here is that we're able to do the selection very early because in very few rounds of selection, because the capillary electrophoresis does such a good job of separating the binding sequences from the non-binding sequences. As we go to later rounds, all it does is really put more demands on the amplification, the PCR side of things. And that if we're going to do more and more replication, more and more PCR, then we have to become more and more careful with our primer design and making sure that we're choosing the right primers and making sure there's not opportunities for misannealing. Then what we did is we went back and looked at the long sequences. These are the sequences that we expect to be having the high affinity, and we wanted to look for abundances of particular sequences in there. And what this slide here shows, if we had a particular sequence of a particular length, what is the chance that it would just randomly be available in that pool? And we can just do this with just basic statistics here, and you say as things get longer, there should be less and less probability of finding of the abundance of that in the pool should get smaller and smaller. And we see that what we actually saw when we actually did sequencing on just random libraries matches us pretty well. What we'll see here is at the longer lengths it kind of plateaus because this is actually representing only finding one sequence in those 30,000 sequences we measured. We can't actually measure something that's less than that. It has to be there. It can't be there less than one time. This slide here is looking at the most abundant sequences we saw at different lengths. And as we go up in different lengths here, this is 15 bases long, what we did is we compared the abundance that we saw in this high throughput sequencing to what we predicted just based on random distributions. And we're seeing we're getting up here about 100,000 times more abundant than we would expect to find just based on random statistics. And so this is telling us that as we go through more and more rounds of selection that yes, we are actually selecting, we are enriching specific sequences here as we're going through the process. Now what was really surprising though is when we started to go through and count the number of sequences that we saw and how many times they showed up in this high throughput sequencing data, they were much less abundant than we expected. So the C1 here, this is the most abundant sequence that we found, and we're comparing the data across all of the rounds here. And we said in pool number 4, out of the 30,000 sequences we measured, this sequence here was only found 199 times. That's less than 0.8% of the population. So that's telling us even though we've gone through this, we've enriched our pool, we're getting high affinity, even the very most abundant sequence we have there is only present at a very low abundance. And so this is why we don't see motifs occur when we're only measuring 30 or 40 clones, because the most abundant one is there much less than 1% of the time. The other sequences there, this is again the most abundant things we found. As soon as we get down to the 8th most abundant sequence, it's there only 4 times out of those 30,000 sequences. So some might worry about this and say, well, we're not enriching very much. So what we did is we compared these sequences over here that we found in high abundance. Okay, these were the same ones from a particular slide. These were the most abundant sequences. And then we used the random number generator, and we compared the affinity for these, and we said, okay, which does a better job, us looking at the most abundant ones or us just picking one that was in that sequence, in all of that sequence data, and only showed up one time. And again, what's really surprising here is we don't really see any difference here. And so what that's telling us is that we're finding thousands and thousands of sequences that look different. But again, when we start pulling them out at random, they all perform about the same. So again, we think this is a big advantage because now it gives us many different lead compounds to basically try in whatever our application is going to be. So just to kind of wrap up here, just some of the advantages that we think that capillary electrophoresis CELEX has. The biggest one is going to be time. There's fewer rounds of selection needed. Again, conventional CELEX, we're talking 10 to 15 rounds. Capillary electrophoresis, very often we're getting high affinity aptamers in 2 to 4 rounds. We have less opportunity for non-specific interactions. We're doing the selection here all in solution. We've never had to do negative selections. We don't get affinity for the capillary wall. And so again, there's less opportunity here for getting these non-specific interactions and having the selections go wrong. We get very few, if any, non-binding sequences. So again, we get all of the sequencing data back, whether it's the cloning or the sequencing, and we can just start pulling sequences out at random. And when we're using protein targets, they all bind our target, and they all bind our target really strongly. We get lots of very different independent sequences. So if you have different, depending on what your application is, if you want to have a sequence that binds in a different environment or binds a particular site on that target, it's nice to have lots of different options to be able to compare, so that way you have more leads to find one that's going to work in your application. We can look at small targets. We can do this in free solution. We don't have to worry about attaching them to a surface. We don't have to worry about having linkers to be able to attach them to a stationary phase. So small targets work just in solution. The capillary electrophoresis is very compatible with modified nucleic acid libraries. If you modify that nucleic acid and start to make it a little bit more hydrophobic, you have a lot of difficulty doing nitrocellulose selections, which selects things out on hydrophobicity. And then also capillary electrophoresis offers new automation strategies, whether it's with commercial capillary electrophoresis instrumentation or moving on and thinking about more microfluidic type approaches as well. So with that, I'd like to thank SciEx again, for the opportunity to kind of speak with all of you. Also like to thank the people who funded this research, the NIH, the National Science Foundation. Also like to thank the group here that's worked on this. Sean Mendoza was the first one to do our IgE selection. Renee Mosing did our HIV-RT selections. Meng Jing did the high throughput sequencing. Jing Yang was doing the small molecule selections against the NMM. And Jay Sung has been more recently working on some of our more challenging targets, looking at membrane protein targets and things like that. And so with that, I'd like to thank all of you for your attention, and I'd be happy to answer any questions that you have.
Well, thank you, Dr. Bowser, for that informative presentation. And as you said, it's time for Q&A. And folks, if you have any questions you'd like to ask Dr. Bowser, please do so now. All you have to do is click on the green Q&A button at the lower left of the presentation window and type in your question into the box that appears on the screen. Click on the send button and we'll answer as many questions as we have time for. So let's get started. And our first question comes from the University of Colorado. Generally, how long are the random DNA sequences that are entered into the selection process and subsequently PCR amplified?
Sure. So the DNA sequences, typically between 80 and 120 bases long. And that's going to be limited by the solid phase synthesis that we can easily get from whatever company we're going to order that from. So again, the library would typically have a 20 base primer, however long random sequence you want to put in, 40 to 60 bases, and another 20 base random primer at the back end to do the amplification.
Thank you. There's a second question comes in from Rutgers University. Could the loss of affinity in the later rounds of the assay for a small molecule binding be due to aggregation of the binders? In other words, in later rounds, are the binders aggregating together and then coming out a peak in a later window than you expected? Do you want me to repeat that, Mike? It was a long question.
I get it. Yeah, that's a very interesting question, and so it's really interesting that we have to think about, especially in early rounds where we're putting in really high concentrations of the DNA or the RNA, there's this opportunity for aggregates to form and DNA-DNA interactions as well as the DNA target interactions, and we have to think about that quite a bit. And those certainly can happen. This is one of the unique and interesting things about the CELEX process, though, is it's not just a single selection round. That you have to be able to have your aptamer survive multiple rounds of selection. And to do that, you have to think about, okay, if you have two DNA sequences come together and then bind a target, that may happen in one round, but what are the chances of those same two sequences finding each other again in the second round? And you have to keep that in perspective with the fact that we have, millions of different sequences, in there that it certainly could happen, but it's probably less likely to happen because there's so much randomness in the pool.
Thank you. This next question comes from Aptogen. Can you select targets in complex matrices like biological fluids?
Sure, excellent question. I don't think I mentioned this in my slide, but one of the really critical things we think when you're doing any selection, any kind of procedure, is to make sure that the binding conditions match your application conditions as closely as possible, whether that's for drug development or diagnostic development. It's one of the advantages of CELEX is that you have a lot of control on what that binding environment is going to be. And so you certainly can match kind of biological conditions when you're using the capillary electrophoresis. Now, one of the trade-offs is that you want to do your binding in something that may be as close to biological as possible, but at the same time you have to balance that with what conditions the capillary electrophoresis can handle. So if you go up to physiological salt concentrations, maybe the current's going to be too high in the capillary electrophoresis. So very often what we do is we try and balance the capillary electrophoresis performance with what we can put in that incubation buffer. So very often we want to make sure that there's salt in there, there's calcium in there, there's potassium in there, but you can also start to add other proteins, other things that are going to be in there that are really critical for your system. And we think that fluidic systems like capillary electrophoresis are really good at adapting to whatever your condition is going to be. You can even go further than that. You can go to non-biological conditions too. If you want to select something in a strange pH or a strange solvent or something like that, that's a really big advantage of CELEX, is it's not tied to the biology, and so you can select under a lot of different kinds of conditions.
Another question's come in, this one from Marshall University. Is there a best way to grow crystals of these, or do or can you determine where they bind?
Yeah, that's always going to be a tricky question. Nucleic acid structures are so much more challenging than protein structures very often. It's hard to do the NMR. It's hard to get the crystals to grow. That's really beyond my expertise, but that's really kind of the steps. If you want to get that picture of what that binding site looks like, that's the kind of things that you have to do. And I would really go and ask an expert in nucleic acid X-ray crystallography about if they have kind of tricks to do that. We haven't worried so much on that end of things. We've worried more on the front end technology development about how to find these aptamers. and kind of letting others kind of go in and kind of characterizing those kind of binding sites. But there's a challenge in the field in general on how to get those binding structures. It's very hard work.
How many rounds, Mike, does it take for PCR that are good? More rounds with less template, or in PCR, or less rounds with more template? And this comes from the University of Kentucky. More rounds with less template, or less rounds with more template?
Right, and so we're kind of limited in this, in that the way we do PCR is different than what most people do with PCR. We're doing very low copy number PCR, and so we don't have the ability to really change how much template we have, because we're amplifying whatever we've collected on the back end of that capillary electrophoresis. In those early rounds, it may only be 10,000 molecules. And so we have to be good at being able to do very low copy number PCR. And then the other thing that's a little bit different is our sequences are quite a bit shorter than most people who are doing PCR amplification. And so we find that we have to change and optimize the conditions just a little bit over regular kind of PCR protocols, just to be able to look at that particular kind of thing. You do have to watch this a little bit as you're going through different rounds, because the early rounds you may not have very much template, and then as you go through more and more rounds you may be getting more sequences back in, and what you might find is that you don't want to use too many, PCR cycles. So in early rounds, very often we have to use quite a few PCR cycles so we get enough DNA. In the later rounds, we'll cut down the number of PCR cycles because we've reached the maximum of the amplification, but all we're really doing is adding more opportunities for misannealing and primer dimers and these kinds of things.
This question comes from ProLab Developments. To date, how many aptamers are you aware of that have been successfully developed using CE-SELEX?
Sure, I don't have a precise number off the top of my head, but it's easily dozens. I would say that there is probably 10 or 20 groups around the world that are actively using CE-SELEX, and that number is going up all the time.
And we're almost out of time, so we have time for one more question. Is how are you doing the fraction collection? Is this done in a separate fraction collector?
No, and so we actually use the commercial capillary electrophoresis instrument to do this. And again, what we're going to do is we're going to see where those non-binding sequences come out, and then we're going to calculate where we expect to find those binders. And we will actually watch the separation taking place, and we will actually basically hit stop on the instrument, tell the robot to change the vials, and either, if the complex is still in the capillary, just to push it into a collection vial, or if it came out before the non-binders, it's already in the vial, and we'll change the vial and wash all the other sequences left on the capillary to waste. And so we'll have an automated system at the back end. Because we're running these separations under sort of non-ideal conditions, the position of that peak can change from run to run, and so it's really important to be able to kind of do that calculation on the fly. If your non-binding sequences come out 30 seconds earlier on one run than the next, then you want to shift that fraction collection, on the fly.
Well, thank you, Mike. We are out of time now, and any of the unanswered questions, we will address them with an email to you. But before we go, I want to let everyone know that today's webcast will be available for on-demand viewing for 6 months from today's live event. You'll receive an email from LabRoots, and they'll let you know when this webcast will be available for replay. So go ahead and share that announcement with your colleagues. They may be interested in this web seminar and had missed it today. So that's all for now, and I'd like to again thank Dr. Bowser for his presentation and SciEx for making this presentation possible. Thank you for joining us, and we hope to see you again soon. Goodbye.